So in summary, what I've shown you thus far, that there is no historical evidence that the secular medical establishment will promote medical research that is consistent with their pro-life values. There is no evidence that cures can be sustainably achieved by the pharmaceutical industry under the current regulatory and financial environment. There is no evidence that the government will take a leadership role in advancing adult stem cell research. And there's no evidence, I haven't gone into the explanation, but there's no evidence that Catholic universities or colleges are going to reprioritize their mission towards focusing primarily on drug discovery, because that's just not what they do. So, the mission of the Institute was created because of this background. So one of the first things that we focused on was to educate people on the medical bioethics of human subject research that is consistent with the dignity of human life. And interestingly, I was once invited to speak to a Protestant church group about, it was sort of a secular versus a pro-life argument about the principles of embryonic stem cell research from the principles of what I just outlined. And after the talk, one gentleman came up to me and said that he was absolutely convinced that there was a moral reason to do embryonic stem cell research. And after he left and listened to me, he said, I am not so sure and confident in my value system. So I realized that there is a segment out there who need to be educated, and they need to sort of question the principles and the issues related to this type of research. And over the years, oftentimes people would find us, they would do their own due diligence, and they would want to support medical research from some cause or another. And they found out that those causes supported fetal or embryonic stem cell research. So they went out and they did their search, looked at the internet, and they came to us and they asked, will you do research for our particular cause? And so, like everything, we start out, like every nonprofit, with very modest beginnings. So we did what we can by trying to bootstrap our resources to do medical research to support as much of a diverse group of diseases as we could. So our objective is to find cures and therapies using adult stem cells, to advance drug discovery and regenerative medicine, and solve problems that are really not adequately addressed in underdeveloped and underperformed areas, such as neurological diseases, those chronic diseases that could benefit from regenerative medicine, like pulmonary disease, rare diseases, and cancer. And it was important to provide innovative solutions that would transcend culture. That is, that would be attractive not just to pro-life, but also to the seculars as well. So how do we differentiate ourselves from other institutions? As people have probably become increasingly aware now, that we are obviously pro-life. We do not support any form of fetal research or embryonic stem cell research or human cloning. What people also may not realize is that we devote a great majority of our research to our funding towards, our private donation funding towards research. So in 2013, we devoted 65% of our our budget towards research and 10% towards education. So to contrast that to some of the more established organizations like the Alzheimer's Association, the American Cancer Society, Coleman's, the ALS Association, they are devoting between 14 to 25% of their budget towards medical research. So despite that they bring in a lot more money, we are putting a lot more of our budget towards medical research. We also have lower administrative overhead. We also are differentiating ourselves by having lower costs and less red tape and bureaucracy. Interestingly, one of the things that I had a conversation with one of the NIH researcher and asked them, what it'll take to get release one of their human cell lines to do medical research. And his response was that essentially would require years of effort and teams of government lawyers to do that. Some people on our board are actually academic scientists, and we ask them how long would it take to get an IRB through their institution, and they say basically you'll be lucky to even get an IRB through. So what we've developed is a very streamlined process, and we work with you guys, the private sector. We work with private doctors, private hospitals, and we can cut through a lot of this red tape. We also do not support an unsolicited research model. In other words, if you look at a lot of the foundations like the American Heart, the American Cancer Society, the NIH, it is they have developed an unsolicited research model. In other words, a researcher has an idea, he submits it, it's reviewed. If they feel it's good enough, it gets funded. But oftentimes, that research is esoteric. It's not designed to identify and accomplish the goals to achieve the milestones to get research that could be approved by the FDA to get a drug into trial. So what we have adopted instead is a solicited research program. That is, we're looking to try to accomplish what are those preclinical milestones that the FDA is going to require, and if we can't do that in-house, then we go out and solicit others who have those skill sets to help bring that research, their research, and try to get those milestones accomplished so we can get our product into the FDA so we can start getting clinical trials. So we have an integrated intramural and extramural program where we have things done in-house, things done outside the house, and we also are differentiating ourselves because we have an integrated clinical program. So a lot of research, for example, oncology research, patients are often the last beneficiary or participants in research from the very outset. So you have a scientist does some research, works in an animal, years later, if something is of novel utility for a patient, they bring that into a clinical trial or as a diagnostic or as a treatment. What we do is that we want to bring the patient to the very beginning as quickly as possible. And lastly, in order to change the drug discovery problems that I just outlined, the Institute has adopted an entrepreneurial biotechnology model. So we're in the business of trying to develop products that can get out into the clinical trials. So our objective is to innovate, to try to reduce the cost and time in drug development. So there's a lot of restructuring over the last 10 years in the pharmaceutical industry. The traditional model where everything can be conducted in a vertical organization in a pharmaceutical company is now broken. So if you look at the basic stages of drug discovery, as I've outlined here, from the very beginning you have discovery, where people are coming up with new molecules, and then it has to go through different discrete periods of development from identifying what drugs might be potential candidates. Those candidates have to be further optimized, and then you have to do the important animal research before you can get an investigational new drug approval from the FDA, and then enter into clinical trial, and then eventually get approved and launched. So the pharmaceutical industry had basically been turned upside down. They no longer have all of this in-house. So everything is increasingly becoming outsourced. And There's also been a downsize in the research and development in the pharmaceutical industry, and a lot of drugs are coming off massive patent expirations. So pharmaceutical companies need novel drugs, they need novel targets, they need access to innovation, they need new intellectual property, and they need to fill these pipelines. So on the far left, we have a variety of different drug libraries, molecules that have been developed by academia, government, the biotech, and there is no idea about the potential utility of those drug libraries. And at the very end, we have the clinical trials. And between those two ends is an area of limited infrastructure where there's a need to be able to take what's already been done and find out what are the best drugs that can be advanced. And so where the Institute's position itself is in that middle. So our expertise is to create disease-in-a-dish cell models and to screen those libraries that either are going to be developed by us or by others to find out the best drug candidates that can graduate. And so in that period, between that period, there is a historical high failure rate and high cost. So what we're trying to do is try to develop a program that tries to optimize this to find the best drugs as quickly, as cheaply as possible.